Assalamu alaikum. Hope all of you are fine. This is our first lecture of the histology of the nervous tissue. In this lecture, we'll discuss the classification of the nervous system, histological structure of a neuron, and how do the neurons they are classified, the types of the neuroglial tissue and their function, microscopic structure of a peripheral nerve, and the microscopic structure of the sensory and the autonomic ganglia, and few of the medical applications. The nervous tissue is composed of the neurons and the neuroglia cells. The neurons, they are the structural and the functional unit of the nervous system, whereas the neuroglia cells and the neuroglial tissue is the supporting tissue of the nervous system, like the connective tissue in the elsewhere in the body. So neuron consists of a cell body which is called the soma or the perikaryon, and numerous dendrites they arise from the cell body. And a single axon arise from the axon hillock and into the axon terminals. So this is the basic structure. The large cell body of the neuron, the soma or the perikaryon, consists of a single centrally placed ovoid nucleus with a prominent nucleolus and finely dispersed chromatin. The cytoplasm of the cell body of the neuron is rich in all the cellular organelles and composed of the rough endoplasmic reticulum, Golgi apparatus, lot of the smooth endoplasmic reticulum, polyribosomes. In addition, the neurons they have basophilic bodies which are called the nasal bodies, and they are the characteristic of the cell bodies of the neurons. The cell body of the neuron, it is surrounded by the numerous dendrites, and a single axon arises from the cell body. The dendrites, they are the multiple elongated processes, and the cytoplasm of the cell body, it extends into the processes of the dendrites. The dendrites, they are basically receive the stimuli from the axons and the other dendrites of the other neurons. The axon they arises from a dilated portion which is called the axon hillock, and this axon they are of a varying length, and they can reach up to the 100 centimeter. The axon hillock is characterized by the absence of the cellular organelles, and in the histological sections, the axon hillock is identified by the absence of the cellular organelles. And the axons, they are responsible for conducting the impulses from the cell body towards the effector organs or the other terminal end, which is the synapse. The neurons, they are classified according to the size and the shape of the processes. So they are classified into the bipolar neurons, the unipolar neurons, and the multipolar neurons. The bipolar neurons, they have the single cell body, and there are two axons which arise from the cell body. Therefore, it is called the bipolar neurons. The examples of the bipolar neurons, they are present in the retina, and also in the olfactory neurons. In the olfactory epithelium, they are also bipolar neurons. Whereas the unipolar neurons, they are also called the pseudo-unipolar neurons. They have a single cell body, and then the axon, a single axon emerges from the cell body, and then it diverges in a T-shaped manner. So therefore, it is called a pseudo-unipolar neuron. So the pseudo-unipolar neurons, they are found in the dorsal root ganglia. The multipolar neurons, they are most abundant in the nervous system, and they have the cell body with the multiple processes arising from the cell body. Therefore, they are called multipolar neurons. They are pyramidal shaped and large cell body, and they are more numerous in the nervous system. So this is shown the pyramidal type of the neurons. which are present in the hippocampus, and they are the Purkinje cells, which are present, Purkinje neurons, which are present in the cerebellum. So the neurons, according to their function, is classified into the sensory neurons or the motor neurons and the interneurons. The sensory neurons, which are also called the afferent neurons, they receive the sensory input and they conduct the impulses from the body towards the central nervous system. Whereas the motor neurons, the efferent neurons, they conduct the impulses from the nervous system, central nervous system, to the effector organs, that is the muscles and the glands. And the interneurons, they are the interconnectors and they establish the neuronal circuit between the sensory and the motor neurons. So they are the internucleal neurons which connects the sensory and the motor neurons. So neurons, they are grouped into the cortex, from which is a six-layer structure in the cerebrum and a three-layer structure in the cerebellum. And the nuclei in the subcortical regions, that is the thalamus, midbrain, brainstem, and the spinal cord. So the neurons, they form the irregular clusters in these areas. The ganglion is a term which is used for the collection of the neuronal cell bodies which are present outside the central nervous system. So they are called the ganglia. The neuron has a basic function of the receiving the stimuli and transduce into the nerve impulse and transfer it to the other neurons. It has also the integrative function, that is, it processes the impulses to the higher centers. And also its a motor function is to initiate the response and impulse to the effector organs. Now, there are structures in the neurons which are called the synapses. The synapses, they are the sites of the impulse transmission, and they convert the electrical signals into the chemical signals. So they are the means of the communication of the neurons. So these are the end of the one neuron synaptic vesicles, which and on the other neurons, they can the synapses, they can be made with the cell body. So they are called the the synapses can be at the cell bodies, they can be at the axons, and they can be at the dendrites. So they can be synapses, they are of the exodendritic synapses, the exosomatic synapses, and the exonic synapses, or the dendritodendritic synapses. The supporting tissue of the nervous system is called the neuroglial tissue. As in other parts of the body, we have the connective tissue, which is the supporting tissue of the organs and the other structures. So in the nervous system, we have the supporting tissue, which is called the neuroglial tissue, and it has the certain cells which are called the neuroglial cells. So it's metabolically and mechanically support the neurons, and it is 10 times abundant than the neurons. So there are different types of the cells which are present in the neuroglial tissue. So in the central nervous system, the neuroglial cells, they are the oligodendrocytes, the astrocytes, the ependymal cells, and the microglia, whereas in the peripheral nervous system, they are the Schwann cells and the satellite cells. So this is the microglia, which is the one of the form of the neuroglial cells, and these are have a cell body with the multiple processes, and they are scattered throughout the central nervous system. They have basically the function of the phagocytosis, which is the similar function of the blood monocytes or the macrophages in the connective tissue. So they have the similar function, and they clear the cellular debris in the nervous system. and they also act as an antigen presenting cells, and they protect the central nervous system from the viruses and the other microorganisms. Now, another type of the cells which are present in the peripheral nervous system, they are the Schwann cells. The Schwann cells analog in the central nervous system is the oligodendrocytes. The Schwann cells, they are responsible for producing the myelin sheaths around the peripheral nerves. So the Schwann cells, they are also responsible for the phagocytosis of the cellular debris in the peripheral nervous system. They are also responsible for the regeneration of the axons. Now, another type of the cells in the peripheral nervous system, the glial cells, are the satellite cells. Now you can see here they're clearly visible around the neurons, the satellite cells. So satellite glial cells, they are the small cells that surround the neurons in the sensory, sympathetic, and the parasympathetic ganglia. So these cells, they help to regulate the external chemical environment, and they are highly sensitive to the injury and inflammation and appear to contribute to pathological states such as the chronic pain. Another type of the cells of the central nervous system, the neuroglial cells, they are the astrocytes. They are very important type of the cells, and their main function is to provide the nutritions. Now you can see here the astrocyte cells. They have the star-shaped structure, like they have a central body and numerous processes. So one of the process, they have the ends which are called the foot ends, and they will be always over the blood vessels. So this is a blood vessel. So they derive the nutrition by their foot ends from the blood vessels, and transmit this nutrition to the other neurons in the central nervous system. So there are basically two types of the astrocytes, the protoplasmic astrocytes and the fibrous astrocytes. The protoplasmic astrocytes, they are found in the gray matter. They have short, thick, and highly branched processes. Whereas the fibrous astrocytes, they are found in the white matter, and they have long, thin, and less branched processes. The ependymal cells. The ependymal cells, they are also called the ependymocytes, and they lines the central canal of the spinal cord and also the ventricular system. So they are basically low cuboidal to tall columnar cells, which are ciliated cells, and they are involved in the creation and the secretion of the cerebrospinal fluid, and they beat their cilia to help to circulate the CSF and make the blood CSF barrier. So you can see this is the central canal, and these are the ependymal cells which are lining the central canal, and you can also see the ciliated appearance of these cells. So they are also thought to be act as a neuronal stem cells. That is the oligodendrocytes. The oligodendrocytes are the cells that produce the myelin sheath in the around the nerve fibers in the central nervous system, and their analog in the peripheral nervous system, as we have discussed, is the Schwann cells. Now, these are all of the neuroglial cells which are shown here. So these are the ependymal cells, which are the tall ciliated columnar cells lining the ventricles and the central canal. These are the oligodendrocytes. The oligodendrocytes, which causes the myelination of the nerves in the central nervous system. The satellite cells, which are the small cells surrounding the ganglia in the autonomic ganglia, the sensory ganglia, and the in the peripheral nervous system. So these are the Schwann cells. This is the nuclei of the Schwann cells, and you can see the Schwann cells wrapped around the nerve fiber. So they produce the myelin sheath around the nerve fibers in the peripheral nervous system. The microglia cells, which are the squamous and have a phagocytic function similar to the macrophages. So this clear the cellular debris in the central nervous system. The astrocytes you can see here with the central body and numerous processes with their foot ends, and they withdraw the nutrition from the blood vessels and distribute these nutrition to the neuroglial cells and the neurons in the central nervous system. So neuroglial cells, they have a mechanical, they provide the mechanical support to the neurons. They prevent the spread of the information in unwanted directions. They have a phagocytic function. They repair the damaged areas by proliferation. That is, they help in the regeneration process, and they also provide the nutrition. Now the peripheral nervous system. So as you know, the peripheral nervous system, it consists of the bundle of the nerve fibers outside the central nervous system, and these bundles of nerve fibers, they are surrounded by the connective tissue sheaths. So main components of the peripheral nervous system, they are the peripheral nerves, which includes the 12 pairs of the cranial nerves and 31 pairs of the spinal nerves, and the ganglia, which are the sympathetic ganglia, parasympathetic ganglia, and the sensory ganglia, and their nerve endings. So together they constitute the peripheral nervous system. Now, histologically, you can see this is a nerve, the fibers. They consist of the axons of the eggs enveloped by a special sheath. So this is called the peripheral nerve. So there are two types of the peripheral nerves which can be identified. They are myelinated nerve fibers and the unmyelinated nerve fibers. So, as we have discussed, that the myelination of the peripheral nerves is carried out by the Schwann cells. So, a single Schwann cell wraps around a single axon and forms the myelin sheath. So you can see here, this is an axon and this is the Schwann cell, the nuclei of the Schwann cell. So it wrapped around the axon, and then it produces the circles around it so that it produces the whorls of the myelin around it. Then the cytoplasm is squeezed so that from it, and the the axon becomes myelinated all around itself. Whereas the unmyelinated nerve fibers, a single Schwann cell envelops the several axons. So the fibers, they enveloped within a simple clefts of the Schwann cells, so that they have the one surface, a surface surrounded by a Schwann cell, and they do not have myelin sheath in one direction. So they are open, and they do not have the myelin sheath here. So this is a diagram showing you the process of the myelination of a peripheral axon. Now this is the diagram showing you the oligodendrocytes and the process of the myelination of the central nerves by the oligodendrocytes. So the oligodendrocytes, they have the numerous processes. They take up the one process take up a one axon and wraps itself around the axon. So, and then when the myelination is completed, it is detached from the process. The cytoplasm is squeezed out. Now, the connective tissue coverings of the peripheral nerves. So, they are covered in the three layers of the connective tissue. So, axon, it is have the surrounded by a connective tissue covering, which is called the endoneurium. So it's a loose connective tissue and it regulates the microenvironment of the nerve fiber. So this is an axon or single nerve fiber which is surrounded by the endoneurium. Then the bundles of the nerve fibers, they are surrounded by the perineurium. So collectively, all the bundles of the nerve fibers, they are surrounded by the connective tissue, which is perineurium, which is a dense connective tissue, and it isolates the neural environment and forms a blood-nerve barrier. Then the whole of the bundles or the fasciculi of the nerve fibers, they are surrounded by the connective tissue covering, which is the epineurium. It's a dense collagen connective tissue with thick elastic fibers. Now, this is a histological section of a peripheral nerve fiber, and this is a longitudinal section, and this is a transverse section. So in a longitudinal section, you can see the bundles of the nerve fibers, and these the nerve fibers, they have the darkly stained nuclei in between them. So these nuclei, they are actually the nuclei of the Schwann cells. In the transverse section, you can see here the connective tissue coverings, that is the epineurium. This is surrounding the connective tissue covering, which is surrounding these bundles of the nerve fibers. And this is the perineurium surrounding the nerve fibers. And then is the inside is the endoneurium, which is surrounding the axons. So these are the central axons, and they are surrounded by the Schwann cells. This is another view of the peripheral nerve. Now the nerve regeneration. So the diseases which affects the Schwann cells, they causes the loss of the myelination, and the demyelination and injuries, they can take place, and it affects the conduction of the nerve fibers. So there can be local changes in the axons, and the or the anterograde changes or the retrograde changes which takes place because of the demyelination, which affects the effect which is due to the affected Schwann cells. Now the ganglia. So ganglia, as we have discussed, they are the ovoid structures, they containing the neuronal cell bodies, and these are supported by the glial cells, which is the connective tissue. So their main function of the ganglia, which are the collection of the neuronal cell bodies outside the central nervous system, is to relay. They function as a relay stations for the transmission of the impulses. So the two types of the ganglia are identified histologically, that is the sensory ganglia and the autonomic ganglia. So this is a diagram showing you the sensory ganglia, and these are the autonomic ganglia. So the sensory ganglia, they consist of the unipolar cell bodies, and they are the cranial or the spinal ganglia, whereas the autonomic ganglia are the parasympathetic and the sympathetic ganglia. They are of the multipolar neurons. Now this is a diagram showing you the section of a spinal cord, and you can identify here the dorsal. At the dorsal root, there is a ganglia which is called the dorsal root ganglia. Now this is the histological section of the dorsal root ganglia, and you can see here the nerve fibers, and these are the neuronal cell bodies in the ganglia, which are the pseudo-unipolar neurons, and these are the efferent nerve fibers, and this is the formation of the spinal nerve. So this is the efferent nerve fiber, on which there lies the dorsal root ganglia with the pseudo-unipolar neurons, and this is the efferent nerve fiber. So the surrounding by the connective tissue sheets, and these are the nerve fascicles which are present here. So now you can compare the multipolar and the pseudo-unipolar neurons in the sensory and the autonomic ganglia. So this is a diagram of the autonomic ganglia. You can see there are the multipolar neurons present in the autonomic ganglia. They are surrounded by the glial tissue, which are the satellite cells, and they have the prominent nucleus and the nucleoli surrounded by the nasal substance. So these are all the Schwann cells. So histologically, we cannot identify the sympathetic or the parasympathetic ganglia. Histologically, we say they are the autonomic ganglia. So the pseudo-unipolar neurons, they are found in the dorsal root ganglia, which are the sensory ganglia, and they are more rounded cell bodies, and they are have the nasal granules also, central nucleus and nucleoli, and they are also surrounded by the satellite cells. But their cell bodies, they are large than the multipolar neurons, and they uh, multipolar neurons, and they are the pseudo-unipolar neurons basically. This is another diagram showing you the pseudo-unipolar nerve cells of the spinal ganglia, and this is the autonomic ganglia of the or the celiac ganglia, showing you the multipolar neurons. Now, the few of the medical applications, that is the brain tumors. The most of the brain tumors, they are the astrocytomas, which are derived from the fibrous astrocytes. The multiple sclerosis is a term which is used in which the myelin sheaths around the axons are damaged by an autoimmune process that affects the activity of the neurons and produce various neurological problems. So this is all about the histology of the peripheral nerve, the ganglia, and the general aspect of the nervous tissue. So next you will study the histology of the cerebellum, cerebrum, and the spinal cord. Thank you.